Is CRISPR used for? CRISPR, or CRISPR-Cas9 as it's sometimes called, is a programmable DNA endonuclease. That means it's an enzyme that cuts DNA, that's the DNA endonuclease part, and it's programmable thanks to a small RNA molecule that goes together with the Cas9 protein and guides it, it's called a guide RNA, guides it to a specific DNA target site with a matching sequence corresponding to 20 specific nucleotides of the guide RNA. Changing the sequence of the guide RNA changes where Cas9 is targeted, that's why it's programmable. When this was first described in 2012, it was a huge deal. The reason it was such a big deal is that the world was primed for it. Not just researchers, but the general public. Back at the end of the 1990s, the Human Genome Project being completed, it created the first published sequence of a human genome. And people, because of this, felt very excited about a coming age of precision genome engineering, going in and fixing the DNA mutations that cause certain genetic diseases. This was all very exciting, but there was one big problem. There were no tools that could actually do that precision genome engineering. So people were very excited in 2009 when there was a new discovery. Something very splashy, but not CRISPR, tails, or transcription activator-like effectors. Tails are proteins from bacteria that essentially hijack the gene expression of the plant host cell that they infect. And they do this because the protein is mostly composed of a large set of repeating units. In each of those repeating units binds one DNA base using a predictable code. And that's actually something unique to tails. And I would know because I spent my whole PhD studying tails, and in particular looking for kind of alternative variant proteins that might have the same properties. And people were very excited about this. There was a lot of money changing hands to invest in applications of this new technology. That was until 2012, when CRISPR was first published as a genome engineering tool. So right away, academic researchers started using CRISPR like crazy as an all-purpose tool for precision genome engineering. They use it to cut DNA, which is a way to generate target mutations, or even to repair those DNA mutations using a repair template, which in the process introduces specific design DNA changes, which could be used, for example, to fix the cause of a genetic disease. This bonanza in the research world is only just beginning to yield tangible real-world application, mostly because of a years-long battle over the patent rights of the technology that was only resolved in 2017. So we're now beginning to see the CRISPR boom, with things like the gene therapy